So it's a pleasure being here, and thanks to the organizing committee for inviting me to present some of the research activities that we've been doing in the lab and how we are getting our hands dirty with some of these massive scale data sets emerging from population scale cohorts. So the one study that I do want to focus on today that is broadly available to most of the research community or any bona fide researcher in the world is the UK Biobank data, which started about less than a decade ago. Just to give you a brief summary for those who are not familiar with the UK Biobank data, this is a population scale research cohort from the UK that's brought together diverse data sources from including health records from hospital and patient record data across 500,000 individuals, verbal questionnaire survey data from approximately 200,000 individuals, physical activity monitor data from approximately 100,000 individuals over the time span of about a week, genotyping data from 500,000 individuals, including array and imputation data, baseline biochemistry data that is due to be released in this quarter, but likely going to be pushed back for another quarter, as usually is the case with most of the research studies. In addition to that, this is one of the largest imaging data sets out there when it comes to combining both the power of genetics and imaging data, with plans for about 100,000 individuals having data from brain imaging to chest and abdomen imaging data, which is continuously being updated in the platforms that they have provided to most researchers. So for anybody who's interested in assessing what type of data is available through the UK Biobank, anyone can go ahead and just search their website, and they have a nice little drop-down catalog that provides some idea as to what data is made available from their research cohort. So the challenges that we faced in terms of analyzing these data sets is that these become quite massive data sets, even for studies that I'm accustomed to being at institutes like the Broad Institute and at Oxford University as well. So here, with these data sets, we're getting to a point of reaching to millions of individuals, having about tens of thousands of measurements across the individuals. And as Nigam pointed out in his talk, right, these research cohorts actually have the timeless compression, whereas in the practice of medicine, you likely will have this time component that will be extremely informative for most of the analysis you do. So that's one of the major limitations from this research cohort. Furthermore, in addition to these phenotype level data, we also have access to genomic data that is on the order of about tens of millions of genetic variants. And as we move into whole genome sequencing, these numbers will likely increase as well. So from our point of view as a small lab starting these research activities, one of the main challenges that we thought about is, well, how is it that we're able to support some of the computing activities and most of the analytics that we want to apply to these data sets? And in fact, telling every student to go ahead and analyze these scale data sets with these orders of magnitude is quite a challenge, especially if it's the first time that you interact with these data sets. So one of the things that we've been thinking about in the lab, and as part of the scientific vision for the lab as well, is going from these big data world to a smaller data set world, where instead of using the raw level data, we started developing models to work on summary level data sets. For example, for each of those phenotypes that we have access to, we're going to run the basic linear regression, basic logistic regressions, and then at the end of the day, you're going to have some summary level data available to you. At the end of the day, the vision is to have methods that apply to these summary level data sets across multiple biobanks or multiple initiatives where we may be able to get access to some of that summary level data. The main limitation, of course, as you may well know from multiple of these biobanks, is that in terms of sharing the raw level data, there's quite strong limitations to doing so. As far as sharing summary level data, the limitations are eased quite a bit. So our methods are built on top of having access to that level of data, the summary level data. And as many of you may know, there are some other biobanks that are soon to be rolled out in the next few years, including an initiative in Finland called FinGen, the China Kadoorie Biobank, and of course the United States initiative called the All of Us program. To that end, what we've been doing in the lab is to develop activities that build on top of other genome sequencing efforts. For example, many of you may be familiar with the EXAC resource, which is a browser that allows you to access summary level data like variant allele frequency or mutation allele frequency across multiple populations. And this has been quite instrumental in changing the landscape of how clinicians or clinical geneticists move forward with some of their research activities. And it's also changed the landscape of some of the biomedical research activities. Here in our lab, we've developed the Global Biobank Engine, and the idea is to support inference across summary statistics, basically assessing association for any given genetic variant across multiple phenotypes that we may have access to. So the first challenge that we came across in designing this Global Biobank Engine was the property that we ended up taking code from the EXAC browser, and that was supported initially in MongoDB as a backend. One of the things that we wanted to do as we were developing statistical models was to perform inference in the backend while also supporting visualization and interactive interaction from the research community with the front end. So that allowed us to, one of the activities that then we moved forward with was thinking about how we design the schemas. For example, we first wrote out what were the levels of metadata that we're going to support, including whether or not a genetic variant was a predicted missense variant in a particular gene, what chromosome that genetic variant is located, the position, reference allele, the alternate allele, and some other metadata that may be available through other openly available resources as well. Furthermore, you also see this level of information that we bring into the schemas, including things like p-values from logistic regressions or linear regressions, and odds ratios from the single variant logistic regression analysis. In addition to that, we also bring in some other level of data like uncertainty estimates or standard errors. For each of the phenotypes, one of the things that we want to do as well is to keep track of the metadata that corresponds to that phenotype, including how many cases we had access to in each of those population biobanks, and what code it corresponds to when we visualize some of the inferences. So one of the activities that became clear that using other database or non-schemaless databases that the challenges we were hitting was doing the inferences while supporting the visualizations. And in terms of seeing the other opportunities or other resources, databases that were available, SciDB became one opportunity that we saw as a real nice treat for using an open source database for supporting some of the activities that we were interested in. In terms of benchmark comparisons to what was available at that moment in time, which was MongoDB, and then comparing that to the second available database, which was SciDB, we saw pretty much quite impressive differences in terms of the database footprint, in terms of thinking about different queries that we were interested in terms of benchmarking. We were interested in counting the number of phenotype associations on a particular chromosome, so that taking a vertical slice of the database. And we saw a pretty nice improvement in speed and performance there. There were other queries that we proposed to them as well as challenges. For example, I'm particularly interested in identifying variants and phenotypes that have effects consistent with protection to disease. So I wanted to see if that was feasible while we were supporting the front-end visualization support in the back end of the database. And with MongoDB, at least, it wasn't feasible, whereas with these new databases, it was feasible. So as I alluded to briefly, one of the main research activities in the lab is to generate effective therapeutic hypotheses from human genomic data linked to health records. The UK Biobank data provides a nice initial data set to work with, as Niga mentioned, there's a lot of challenges in how you interpret health records, and in fact, most of them come quite noisy and quite, a lot of processing needs to happen under the hood. And as a geneticist, those are activities that I'm not familiar with, but I reach out to my colleague Nigam for help with that, especially as we start bringing in new data sets like primary care level data that they'll soon start releasing. So for those that are not in the field of human genetics, genome-wide association studies has been the primary discovery engine for that field. The idea is basically that you look at genetic mutations sparsely populated across the genome and assess for some level of association to disease. These activities, or genome-wide association studies, have been quite successful in identifying regions of the genome that are associated to disease. Typically, what you show in terms of a study that moves forward with GWAS is this plot called a Manhattan plot. On the y-axis is the minus log 10 p-value, and on the x-axis is the location of each of those genetic variants that you assess for association. And for most diseases, we've been quite successful identifying regions of the genome that have some non-zero effect corresponding to that disease. So you may ask yourself, why was there an asterisk after successful in the last slide? Well, one of the reasons is that for these genome-wide association studies, most of the associations that have been identified have the property of having very weak effects, which then introduces some limitations how you move forward with the interpretation of that genetic finding. In the context of type 2 diabetes, for example, all common variant associations were tiny. There maybe was one exception, TCF702, but for the most part, the majority of them were quite tiny, very close to 1 in terms of effect sizes. Now, as we move from common variant association studies to rare variant association studies, that is, association studies that assess genetic studies that assess for association of rare genetic mutations to have any role in disease predisposition, we started seeing the pattern of mutations that had a bit of a more modest or stronger effect. In fact, one of the properties that started emerging as well were a few examples, a couple of examples, I would say, of mutations that instead of increasing risk for disease, showed effects consistent with protection to disease. And this became quite exciting for most geneticists in terms of thinking about, now that you have access to whole genome sequencing data, or at least the complete picture of genetic variants across the frequency spectrum, can we identify mutations that may have better insights as to how we move forward with developing therapies for disease? So to that end, in the lab, we've been developing algorithms and statistical models for that moment in time when we do have access to whole genome sequencing or exome sequencing data across very large sample sizes. Again, by definition, these genetic variants are rare, which means that in order to have enough information to assess for any evidence of association, you will need very large numbers. So to that end, we've been developing methods, statistical models called for rare variant associated studies across multiple phenotypes. And the idea is that by design, we will work on these models that operate on top of summary level data as opposed to raw level data. Because at the end of the day, having access to the raw level data will be a very daunting task. So we've put out a bioRxiv preprint that describes this first statistical method approach that we've developed. In addition to that, we've also posted some applications to the UK Biobank dataset and the rare variant content that is included in the UK Biobank array. In the context of glaucoma, for example, we've identified genetic associations that are consistent with protection to glaucoma in a gene that's strongly expressed in the vitreous humor, and very nice properties of multiple alleles showing effects consistent with protection to the disease. Now, for us, when we initially started the application of these models to the summary level data, we didn't know what to anticipate. I could tell you initially with the development of tools used in human genetics like Plink and PlinkSeq, in Plink, when we started developing PlinkSeq and we started looking at rare variant association studies, running a whole genome scan or exome-wide scan generally took on the order of hours in order to move forward with an entire gene scan. By using summary level data, by having a schema that operates quite nicely and a database that operates quite nicely on the schema of summary level data, we're able to stream these models while people are browsing the features supported in the Global Biobank engine in approximately 10 seconds, which is quite impressive, at least as a first-time user of these approaches. So it really speaks to the utility of thinking about efficient database design and these types of schemas that may help us as users of these databases move forward with our scientific activities. So to that end, in terms of more recent research activities that we move forward with, recently we published a paper in a journal that focused on these protein truncating variants across the entire phenome, and we've identified multiple mutations that confer risk and protection to disease. This was highlighted in Genome-wide as a nice proof of principle as to how you can combine these data sets to improve our understanding of the genetics of disease. Just to highlight a few examples that we highlighted in this paper, we identified genetic variants that completely truncated the protein of IL-33, and we identified strong evidence of association and conferring protection against asthma. The signals of association were consistent with decreasing eosinophil counts and decreasing asthma risk. Furthermore, truncating variant in IFIH1, this signal had been reported by a collaborator in Cambridge University at that time, who identified loss of function mutations that confer protection against type 1 diabetes. And in our study, we showed associations of these genetic variants beyond type 1 diabetes, showing that it had effects consistent with protection against psoriasis, hypothyroidism. However, it also had effects consistent with increasing risks to diseases like inflammatory bowel disease and Crohn's disease, and ulcerative colitis. So the next challenge that comes into play in these flavors of studies is what you do when you scale up these rare variant association studies. I've touched base on some of the research activities that looked at a few limited number of rare variants that were included in the array. But recently, there's been an announcement of large investments in moving forward with sequencing of the entire population cohort. So we will have access to all rare genetic variants across these 500,000 individuals that we will be able to scan for these types of signals. So in the last 5 minutes, what I want to do as well is to revisit some support of the additional features in the Global Biobank engine, including multiple views of analysis, right? One of the things that we're interested in communicating to the research community is how effects or genetic associations across multiple diseases correlate with each other. In addition to that, how specific HLA alleles associate with diseases and what their effect sizes are. In addition to that, for a specific genetic variant, if you're interested in a specific disease, you can also assess its association to multiple other diseases, these PHEWAS-like pages. And browsing these data is largely influenced or affected by the performance of the databases that we put under the hood. And there's many more activities that we're moving forward with more advanced statistical modeling techniques, for example, including how we use genetic data to improve our ability to quantify risk of a disease to an individual. So finally, just to wrap up, for these activities, it's been quite critical to consider our backend database that we've used, and especially as we start considering scalability, security, and versioning. With that, I'd like to thank all the members of the lab who've contributed to these research activities, and all my colleagues who've also helped out in discussing some of the issues that may come about as well. All right, thank you. Perfect. So the client tool being the visualizations. Yeah, so great question. So for that we've used Plotly. You know, Plotly has been phenomenal. Plotly. Uh, yeah. Yeah, yeah. It's been quite easy. You know, the first visualizations were I used to write some D3 code, and it took quite some time. And then with Plotly, it was quite easy to then add some visualization, some interactive bar plots that allowed us to communicate what was going on under these summary level data sets that we're coming up with. Okay.
Question?
So for these data sets, you said that you sometimes use MongoDB or SciDB on the back end to do some analysis. Are these in like Stanford's own data center, or do you actually use the cloud anytime to do any of these types of processing?
Oh, great question. So, you know, MongoDB, the reason why we started off with MongoDB was because the legacy code base from Exec and the IBD browser that Somali mentioned. Given that it's summary level data, we use the Google Cloud instances here to support the server. And then we move forward with replacing it with another database. So thus far, in terms of using the summary level data, it's been quite impressive. You know, we haven't looked at what we can do in order to support querying across the raw level data, right? You know, that needs, I would imagine that probably needs some custom code as well. For example, there are efficient, so Plink 2.0, for example, is a tool that's developed to efficiently query genetic data. Whether or not there are database solutions out there that can query that database, that data structure in an efficient manner that competes with Plink 2.0 is unclear to me. So, you know, some of these data types will need special handling. The temporal data will probably need some special handling. Nigam alluded to some data that really did need some special handling, and the genetic data is one of those flavors as well. The summary level data is, I would say, it's probably you know, a bit easier to represent in a schema, but the opportunity to do both the querying and streaming of algorithms was something that that was quite attractive to us as users. Yeah. Okay, good.
I had
Yes.
one question. So in terms of the UK Biobank data, are you just looking at the genetic data, or are you looking at the other clinical data that's available as well?
Yeah, so you know, one of the main limitations of the UK Biobank data is that the clinical data is not very rich. The verbal questionnaire data is impressive. It's very similar to what I would imagine 23andMe does. Self-reported. The clinical data is limited to hospital inpatient record data. There is supposed to be a release of new data that includes more, you know, primary care level data, and that's supposed to be richer and represent more of this long trajectory of collection of the diagnoses for individuals. So if you wanted to study a disease like type 2 diabetes, for example, the hospital inpatient record data would be quite useless, whereas the verbal questionnaire data becomes quite informative. So there's, you know, it does have its strong limitations, the UK Biobank data. However, I think it's at the scale that we're talking about, it's one of the nicer data sets that is out there that is available to researchers and is designed as a research cohort. So, yeah. All right. Thank you.
Thank
Appreciate
you.
it.